Hello, we are at the International Scientific Conference on Environmental and Climate Technologies. My name is Kārlis Valters. This is an episode of the podcast Sustainable Worlds, and I'm pleased to have with me Yagut Allahverdiyeva Rinne from Finland, who just had a very interesting presentation about algae photosynthesis. Would you please tell about your research topic?
Yes, I'm professor at the University of Turku, and my background is on photosynthesis. So my group is studying photosynthesis, how this natural process occurs, and fundamentals to understand how photosynthesis occurs, and also how it can be applied in real life. We are very much willing to improve photosynthesis because this is the key for sustainability. So we have two different directions. One is to understand, to do fundamental research. The other direction is that to bring photosynthetic organisms to industrial applications.
Right. So photosynthesis has been evolved during evolution, so one might think that it is perfect already, but you're saying now that that can be enhanced in some way. How?
Yeah, that's correct. I mean, photosynthesis is perfect, but it is more about the nature. So photosynthetic organisms they evolved to survive under different environmental conditions. That's why they have many different regulation mechanisms. So they try to, if there is a more light, for example, that can be stress for the cells. If there is a drought stress, it's a stress. So they try to arrange photosynthesis so that it doesn't collapse. And that's why this regulation needs a lot of energy. Regulation needs a lot of extra electrons. So this becomes, in a way, if you are thinking that using the photosynthetic organisms for bioproduction, you are wasting certain energy for regulation. So in natural condition, this regulation is important. But if you have a controlled industrial condition, then this is the waste. So that's why we are trying to understand where is the waste points of the photosynthesis, and then to cut down that waste points to make it more efficient. So theoretical conversion efficiency, light to product conversion efficiency of photosynthesis is about 10-12%, which is good enough for industrial application, but in nature this is around 1%, less than 1%. So there is a huge gap. I mean, we have a possibility to reach the theoretical maximum.
But there's a lot of sunlight, even if it's just 1% used.
Yeah, there is a lot of sunlight, but they have to use it more efficient way, not to waste the sunlight. And also they use the only visible light.
Right.
So there are also quite much light. For example, far-red light is lost. So the one thing is that, for example, expanding the photosynthetic absorption to far-red light. So this will also help to increase the light-to-product conversion efficiency.
So chemically, what happens in photosynthesis? Basically, CO2 is transformed to oxygen, right? There's many other biochemical reactions going on, right? What kind of organisms do you use?
So during photosynthesis, actually, the light energy is captured, and that light energy used to split water to oxygen, protons, and electrons. And this oxygen, what we breathe, is coming from photosynthesis. But this electrons coming from water is used to fix atmospheric CO2. So light energy at the end is needed to fix the CO2. Regarding the organisms, I have a background on plants, but when I started to think about more applied part, we slowly moved to microalgae and cyanobacteria, because we think that they have a more capacity to be sustainable for bioproduction, biorefinery, bioeconomy. Of course, plants are important. They are crop, so we need also to improve the photosynthesis for efficient crop production. But it is much easier to learn with cyanobacteria and algae how to improve photosynthesis and then transfer this to plants, because the lifetime, of course, the plants, cyanobacteria can divide in 2 hours, but for plants you need a quite long time.
Right. So this might not be exactly your research topic, but it might be interesting to the listeners that cyanobacteria, what we usually often call algal blooms in natural waters caused by man-made nutrients from agricultural runoff, etc., from detergents and etc. They're called algal blooms, but they're actually cyanobacteria, right?
Yes. Yeah. I mean, if you think about taxonomy, then cyanobacteria and algae, they are different things. So cyanobacteria is the prokaryotic organism. So they are,
Prokaryote is
they don't have a separate nucleus
in the cells.
In the cells, but in algae they are eukaryotic, so they have a nucleus separately, and they have a different organella. So it's a more complex.
Yeah.
So, however, in biotechnology, for the sake of the simplicity, usually it's called algae sector and microalgae, which also covers cyanobacteria.
Right.
Yeah, but the blooms, yes, it's mostly cyanobacterial strains. It can be nostoc, it can be microcystin, and there are quite much toxic ones also. I think that scares people.
Plus they rot and they consume oxygen in water.
Yeah, and they use a lot of, if there is a lot of nitrogen and phosphorus eutrophication, then summertime they start also to grow. There are quite much work also done that what is the exact reason for blooming. I think it's not very clear, but still it's a quite important part still of the research ongoing all over.
Very interesting. So just to explain, the taxonomy, it's a way to classify the organisms. Like this would be algae, this would be bacteria or yeast, etc. So there is a very rich world on the micro level that we don't see with the naked eye,
Absolutely,
right?
yes.
It's very interesting to study, isn't it?
Yeah. There are biodiversity amazing. So biodiversity of the algae cyanobacteria is amazing, and we have studied only a very small amount. In the labs we are stuck to this few strains, but if you go to biodiversity, you can find, for example, there are some red algae which can grow at pH as low as 2, for example. pH 2 is quite normal for them to grow, or some cyanobacteria or algae can bloom under in the Arctic. If you can see there, for example, there is a rose color of the pink color of the snow, and this is the Chlamydomonas blooming under the snow, and snow become a pink color. And that color comes because this Chlamydomonas, because of this temperature and light combinations, they start to produce the antioxidants, for example, and that's why the color is there.
And then there are even microorganisms that gain energy on the bottom of the oceans from the geothermal energy, not even from the sun.
Yes, that's correct, yes.
And we need to wrap it up now. Would you please tell briefly about the consortium of the Nordic universities, 10 universities in this project you talked about? How do you cooperate and specialize?
We had the Nordic Center of Excellence, Nordaqua, and that was funded by Nordforsk by economy program, and the main idea was to bring algal sector close to industrial applications in Nordic region, considering Nordic climate. And we were 10 different universities from 3 different Nordic countries, and also we had 10 societal partners. And that was a quite actually big consortium, and about 6 years we operated. Now consortium is over, but still we have a very strong links, and we are applying together to different projects to continue that project. Yeah.
So scientists from different research centers cooperate, bring their own expertise.
Yes.
Okay, thank you very much. Our guest today was Professor Yagut Allahverdiyeva Rinne from University of Turku. Thank you for an interesting discussion. This was an episode of the podcast Sustainable Worlds. My name is Kārlis Valters. Thank you.